Hello, it's Shudena Nagar. Today we're going to be solving October-November 2020 Paper 1 Variant 3. And before we start, I'd highly appreciate it if you would take a moment and subscribe and like this channel. I highly appreciate your support. As a gift from our channel, we're going to be releasing free Topic 1 PDF notes. All you have to do is send us your email in the Google form found in the description box down below. Number 1. What are the appropriate units for measuring diameters of alveoli, diameters of white blood cells, and the width of cell walls? Diameters of alveoli we all know is going to be in micrometers. Diameters of white blood cells, we know that the diameter of the white blood cell is always going to be 7 micrometers. And the width of cell walls we also know is going to be 7 nanometers. Therefore, D is going to be the correct answer. The actual diameter of a prokaryotic cell is 0.5 micrometers. An electron micrograph of this cell has a magnification of 50,000. What is the diameter of the cell in the image? As we all know, the equation for magnification is IM. And here it's asking for the image size. And here we have the actual size and the magnification. First of all, we must convert the units from micrometers to millimeters. We do this by dividing the answer by 1000. Then we multiply the actual size by magnification. This gives us C. 3. The electron micrograph shows some cell structures. Some cell structures are listed. Which cell structures can be seen in the electron micrograph? Clearly, this black spot indicates that nucleolus is present. 2. Chloroplasts. Here, chloroplasts are not visible. That's what chloroplasts would look like, an oval-shaped large structure. So they are not present. 3. Microtubules. Here, microtubules are not really visible. 4. Ribosomes. As we could see here, there are granules throughout the cytoplasm. These granules are definitely the ribosomes. 5. Mitochondria. Here we can see the mitochondria. Therefore, it is present. 6. Plasmodesmata. I don't know if you could see it clearly. However, plasmodesmata are found in the holes in the cell wall. The function of plasmodesmata is to allow the movement of substances between plant cells. Therefore, the answer is going to be B. 4. Which part of the cell is often continuous with the rough endoplasmic reticulum? Here, I've included a diagram to make it easier for you to visualize. As we all know, the rough endoplasmic reticulum is continuous with the nuclear envelope. Therefore, the answer is going to be D. Which statements about ATP are correct? 1. It is produced in the chloroplasts. This is correct by Calvin cycle. 2. It is used during protein synthesis. Yes, ATP is used in order to synthesize proteins, both in transcription and translation. 3. It contains deoxyribose. Deoxyribose is incorrect. It actually contains a ribose sugar. Deoxyribose is present in the nucleotides in DNA. So I repeat, in DNA nucleotides. 4. It is used in facilitated diffusion. As we know, facilitated diffusion is the movement of substances through transport proteins down concentration gradient. Therefore, it is a passive process. Therefore, it does not require ATP, so it is incorrect. 5. It is used to load sucrose into companion cells. This is correct because ATP is used in proton pumps where they pump H+ ions out of companion cells. Therefore, the answer is going to be A. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. 1. mRNA passes through to the ribosome. As we know, it passes through the nuclear envelope in order to leave the nucleus. 2. Organizes microtubules to produce the spindle during cell division. As we all know, it's the centrioles found in the centrosomes is what organizes the spindle fibers during cell division, particularly in prophase. 3. Synthesis of lipids. We all know that the smooth endoplasmic reticulum is responsible for the synthesis of lipids and steroids such as cholesterol, which is used in cell membranes. Therefore, the answer is going to be D. Going back to the letters, X is a double membrane with pores, and this is the nuclear envelope. Y, non-membrane bound cylindrical structures. As we all know, microtubules are cylindrical structures. Therefore, Y is going to be correct. V is membranes which surround an enclosed inner cavity. This enclosed inner cavity are tubules, and these tubules is what forms the smooth endoplasmic reticulum. A student carried out tests for biological molecules on the sample of milk. The test and the results were as follows. Which biological molecules must be present in milk? So let's start with the first point. Here it says here to Benedict's solution. Benedict's solution is used to test for the presence of reducing sugars. And it gave a red color. So from blue to red is a positive result. Therefore, reducing sugars must be present. Now, boiling with acid followed by neutralization, then heating with 80 degrees Celsius with Benedict's solution also gave a red color. Here, we cannot guarantee that adding the acid, by the way, if we add the acid, we hydrolyze the glycosidic bond of a non-reducing sugar, forming a reducing sugar, and this is the basis for a non-reducing sugar test. Since we cannot see a color change, here it's red and here it's red, we cannot at this point guarantee that a reducing sugar is present. This is only going to be correct in one case, and it is if here, for example, it gave a yellow color, then here it gave a red color. It went darker means that there might be a non-reducing sugar, but in this case we cannot actually guarantee it, so non-reducing sugar is going to be incorrect. The third point, adding Biuret's solution gave a purple color. Now, Biuret's solution tests for the presence of proteins, and purple color, so color change from blue to purple, is a positive result for proteins, so protein is present. And the last point, adding iodine solution gave a yellow color. As we all know, iodine solution is yellow, and a positive result for starch is going to be blue-black color. Here, there hasn't been a color change, therefore starch is not present. In this case, 2 and 3 is correct, and the answer is going to be question number 8. Which row is correct for carbohydrates? We have monomer, polymer, and macromolecule. For monomer, here we have fructose. Yes, it is a monomer. Glucose, also it is a monomer. Monosaccharide is a monomer, and sucrose is a disaccharide, so it's not a monomer. Polymer is a large molecule made of many repeating units. In this case, amylose is correct because it's made of many repeating units of alpha glucose monomers. Deoxyribose is also a monomer, so it is incorrect. Glycogen is a polymer also made of alpha glucose subunits. And starch is a polymer made of amylose and amylopectin. Now moving on to macromolecule. Macromolecule is basically a giant molecule made of many monomers, and they don't have to be repeating. Let's start with cellulose. Yes, it is a macromolecule made of thousands of beta glucose monomers. Amylopectin is also a macromolecule made of many repeating units of alpha glucose. Deoxyribose is a monomer, so it's incorrect, and glycogen is correct and also made of alpha glucose repeating units, as we mentioned earlier. Therefore, the answer is going to be A. 9. Which molecules contain 1 to 6 glycosidic bonds? So the molecules which contain 1 to 6 glycosidic bonds are going to be highly branched molecules. Let's start with amylopectin. Amylopectin is made of both 1 to 4 and 1 to 6 glycosidic bonds. So this is correct, and amylopectin joined with amylose forms starch. 2. Glycogen. Glycogen is also made of 1 to 4 and 1 to 6 glycosidic bonds. 3. Starch is also made of 1 to 4 and 1 to 6 glycosidic bonds. The reason for this is amylopectin contains both of these bonds. Therefore, all three of them is going to be correct, and the answer is going to be A. 10. Which row correctly shows the ratio of carbon atoms to hydrogen atoms for glucose, a saturated fatty acid, and sucrose? So let's start with glucose. We know that the formula of glucose is going to be C6H12O6. Therefore, it's obvious that the ratio is going to be 1 to 2, as seen in the general formula. Saturated fatty acid, here I've included the formula of a saturated fatty acid, and it is obvious it's also going to be 1 to 2. For sucrose, it's a disaccharide, and I've here included the general formula. So simplifying the ratio gives us 1 to 1.8. Therefore, the correct answer is going to be A. Which row shows a correct comparison between a phospholipid molecule and a triglyceride molecule? Phospholipid molecule contains a hydrophobic phosphate group. Hydrophobic is incorrect. It's a hydrophilic phosphate group that is able to form hydrogen bonds with water. B. Contains one glycerol molecule. Yes, this is correct. It does contain one glycerol molecule and 2 fatty acids. So this is the glycerol, 2 fatty acids, and 1 phosphate group. C. Fewer ester bonds than triglycerides. As we all know, the structure of triglyceride is 1 glycerol and 3 fatty acids. And as we all know, the fatty acids are joined to the glycerol by an ester bond. Therefore, here the fatty acid has 3 ester bonds, and because here we have 2 fatty acids, it's going to be 2 ester bonds. So, phospholipid molecule does have fewer ester bonds. This is correct. D. More fatty acids and fewer fatty acids. As we all know, this is incorrect. And the correct answer is going to be C. 12. Adult human hemoglobin typically consists of 2 alpha chains and 2 beta chains. Approximately 5% of humans have one amino acid in the beta chain that has been changed. This change affects the structure and stability of hemoglobin. Which levels of protein structure could be affected in the hemoglobin of the humans with the changed amino acid? So here it says it changed the amino acid. Changing the amino acid is definitely going to be the primary structure also altered. The reason for this is the primary structure definition is the sequence of amino acids. So we know that the primary structure is altered. Then, if the primary structure is altered, we all know that every amino acid has different bond interactions. So changing the primary structure changes all these other bonds too and its interactions, therefore changing the structure and stability of hemoglobin. Therefore, the answer must be D. Lysozyme occurs in human tears, saliva, milk, and mucus. Lysozyme has a role in the immune system and hydrolyzes peptidoglycan. Which description of lysozyme is correct? So, to start with, peptidoglycan is a component of cell walls found in bacteria cells or prokaryotic cells. Now, A. It is an extracellular antibody. antibody is incorrect, it's an enzyme. C. It is an extracellular antibacterial protein. Yes, it is a protein because enzymes are proteins. And it's extracellular because it says it occurs in human tears, saliva, milk, and mucus. And these are secreted by glands, so it's extracellular. And the answer is going to be C. Which letter in the flow diagram shows the effect of adding a competitive inhibitor to an enzyme catalyzed reaction? It is a reversible reaction. This is correct, it is a reversible reaction. The reason for this is if we add more substrate to a competitive inhibitor solution, in this case, the increased amount of substrate outweighs the competitive inhibitor. Therefore, it is a reversible reaction as long as we increase substrate concentration. Now, affects the Vmax value of the enzyme. As we all know, because it is a reversible reaction, the Vmax will not be altered as long as we increase the substrate concentration. B is going to be the correct answer. No, it does not affect the Vmax, contrasting a non-competitive inhibitor which actually does alter the Vmax. The cell surface membrane structure is described as fluid mosaic. Which statement describes the mosaic part of the cell surface membrane? Let's start first describing what fluid means. Fluid means that phospholipid molecules are able to diffuse throughout the cell surface membrane and move freely. Mosaic means the scattered protein pattern. Therefore, let's start with A, the different patterns that are obtained by the moving phospholipids. No, the patterns are obtained by proteins, not phospholipids. B, the random distribution of cholesterol molecules within the phospholipid bilayer. No, it has nothing to do with cholesterol. C, the regular pattern produced by the phospholipid heads and membrane proteins. No, they do not have regular patterns. D, the scattering of the different proteins within the phospholipid bilayer. This is correct, and the answer is going to be D. Question number 16. Solute X is at a higher concentration outside a cell than in the cytoplasm. Which processes may allow solute X to be moved through the cell surface membrane? So, here we have a cell, and solute X is at a higher concentration outside the cell. So it will go inside the cell, down concentration gradient. So let's start with A. Active transport is the movement of solute particles through the cell surface membrane through transport proteins. But in this case, it's against concentration gradient, so let's just leave it till the end. Now, let's look at diffusion and facilitated diffusion. Diffusion is movement of substances down concentration gradient, so it's correct, and facilitated diffusion is the movement of substances also down concentration gradient, but only through transport proteins. So this is correct. B. Active transport, diffusion, and osmosis. Osmosis in all three of these are incorrect. The reason for this is osmosis is the movement of water molecules down water potential gradient. But here we're talking about the solute. Therefore, the only correct answer must be A. The 3 molecules found in prokaryotes are listed. Which molecules are found in prokaryotic cell surface membranes? So let's start with phospholipid. Yes, all membranes contain phospholipids. 2 proteins. Yes, the proteins are present, either embedded, intrinsic, or extrinsic proteins. 3. Peptidoglycan. Here it's talking about cell surface membranes, not cell walls. Peptidoglycan is a constituent in cell walls of prokaryotes. Therefore, this is incorrect, and the answer is going to be B. What happens to the surface area to volume ratio of a cube when the length of each side is doubled? Here I've decided to include an example for you to make it easier to visualize. Here I've worked out the surface area to volume ratio, and it's going to be 6:1 to the simplest ratio. Here I've doubled the two sides and acted upon that. Here it gave me 24:8 surface area to volume ratio. As a simplified ratio, it gave me 3:1. Here we could clearly see that the surface area to volume ratio has been halved. Therefore, the ratio decreases by half and the answer is going to be B. 3 parts of a chromosome and their functions are listed. Which part is matched with its correct function? Let's start with P1 and F2. P1 is centromere, F2 holds 2 chromatids together. Yes, the centromere holds the 2 sister chromatids in a chromosome, so this is correct. B. P2 and F2. P2 are histone proteins, F2 holds 2 chromatids together. This is incorrect. Histone protein actually holds the coils of DNA together, makes it more compact, and allow it to fit in the 10 micrometer nucleus. Because the DNA strand is actually 2 meters. So it has to condense into that 10 micrometer nucleus by wrapping it around histone proteins. P2 and F3. P2 is histone proteins and F3 prevents loss of genes. This is incorrect. This is actually telomeres, which prevents loss of genes. T. P3 telomeres and F1 telomere, as we mentioned, it does not hold the coils of DNA, so the correct answer is going to be A. Which features of mitosis are important for a single-celled organism? 1. Asexual reproduction. Yes, the cell has to reproduce, and the only way to do this is by asexual reproduction, so mitosis is vital for it. 2. Production of genetically identical cells. Yes, when asexual reproduction takes place, obviously genetically identical cells are going to also occur. Therefore, this is correct. 3. Here it says growth. Growth in this case is incorrect because an actual cell cannot grow by mitosis. It only divides by mitosis. What grows are tissues. When cells divide, more cells of this tissue is provided, therefore the tissue is what grows, but not the cell. Therefore, the answer is going to be B. Some events in the cell cycle are listed. Which events are part of mitosis? So, as we know, mitosis is P, mat, prophase, metaphase, anaphase, and telophase. Therefore, metaphase is the only correct answer, and the answer is going to be D. Which row represents the correct features of guanine? As we all know, that guanine is a purine, which is a double-ringed structure. Therefore, C and D is going to be correct. And is a purine? Yes, it is a purine because it's a double-ring structure. Joint is complementary base via 3 hydrogen bonds. As we all know, cytosine pairs with guanine via 3 hydrogen bonds, and adenine pairs with thymine via 2 hydrogen bonds. So this is correct. And pairs with thymine, as I just mentioned, it pairs with cytosine, not thymine. So this is correct. And the answer is going to be C. 3 bacteria were grown in a medium containing a nitrogen-15 isotope. After several generations, all of the DNA contained N15. Some of these bacteria were transferred into a medium containing the common isotope of nitrogen N14. The bacteria were allowed to divide once. The DNA of some of these bacteria were extracted and analyzed. This DNA was all hybrids containing equal amounts of N14 and N15. In another experiment, some bacteria from the medium with N15 were transferred into a medium of N14. The bacteria were allowed to divide 3 times. The DNA of some of these bacteria were extracted and analyzed. What is the composition of this DNA? I'm going to start with N15 as mentioned in the question. And now it is allowed to divide 3 times. Therefore, in each of the 3 times, we were adding N14 isotope to it. So in the first division, it's going to form N15, N14, and N15 and N14 because we're adding N14 to each of these. Now, for the second division, and as it is mentioned here, all the DNA was hybrid. So this is hybrid DNA containing both N15 and N14. Now, in the second division, we're also adding N14 to these, forming N15, N14, N14, 14, N15, N14, and N14, N14. Here, as we can see, there is 50% of N14 and another 50% which is hybrid. In the third division, we're going to also add N14 to all these. So N15, N14, Now, in the third division, we could clearly see that now we have 6 N14, N14, and here we have 2 hybrids. Therefore, if we divide the simple ratio, 6 out of 8 for N14 is going to give us 75%, and 2 out of 8 is going to give us 25%. For hybrid. Therefore, the only correct answer in this case will be C. In theory, what all the students do is must memorize the values. However, I've answered it in depth with you in this question. A molecule of tRNA has the anticodon sequence of UAC. What will be the corresponding nucleotide sequence in the DNA? So, a general rule of thumb, the anticodon or the codon sequence in both transfer RNA and DNA are going to be the same, but we have to swap the uracil with thymine because DNA does not have uracil. Therefore, if the anticodon sequence in tRNA is going to be UAC, then the nucleotide sequence in DNA is going to be TAC. We've only swapped the uracil. In this case, the answer is going to be 65. Some of the features present in transport tissues in plants are listed. Which of these features are present in xylem vessel elements? 1. Lignified walls. This is correct. Xylem vessel elements walls are lignified for structural support. 2. Cytoplasm. Cytoplasm is incorrect because we know that xylem vessel elements are hollow tubes or dead cells. Therefore, they cannot have cytoplasm. 3. Many mitochondria. This is incorrect because, as we mentioned, they are dead cells. 4. Pits. Pits is correct and they allow lateral air movement of air bubbles outside the xylem vessel element. And pits are formed in the place of the plasmodesmata. 5. Plasmodesmata. As we all know, the definition of plasmodesmata is strands of cytoplasm, and we just mentioned that xylem vessel elements do not have cytoplasm because they are hollow cells. Therefore, this is incorrect. Sieve plates. Sieve plates are only present in phloem, sieve tube elements, not in xylem vessel elements, so this is also correct. The only correct answer is 1 and 4, and it's going to be B. 26. The diameter of a tree trunk usually decreases slightly during the day and increases slightly at night. Which 3 environmental factors, if increased, could cause the largest decrease in diameter? So let's break down the question. Why does even a decrease in diameter happen? At the day, the rate of transpiration increases, so more water molecules are removed out of the plants through the leaf through the stomata. Therefore, there is an increased transpiration pool. During the day, if there's an increased transpiration pool, this means that more water molecules are going to be moving up at a faster rate. Therefore, we know that water moves by both adhesion and cohesion. In this case, the cohesion forces increases between water molecules as they move up at a faster rate. When the cohesion forces between water molecule increases, this creates a tension force which pulls the diameter of the stem inwards due to the tension force. Therefore, here we're trying to look at the factors that actually increase the rate of transpiration. In this case, we know that D is going to be the correct answer because humidity is incorrect. The reason for this is that humidity actually lowers the concentration gradient, and lowering the concentration gradient lowers the rate of transpiration. So D is going to be the only correct answer. Sucrose moves within a phloem sieve tube from a source to a sink by mass flow. Which type of gradient within the phloem sieve tube between the source and the sink causes the movement of sucrose? So let's break it down. As we all know, when sucrose moves from companion cells into the phloem sieve tube elements, the concentration of sucrose here increases. There is a higher concentration of sucrose. Therefore, water from adjacent cells goes into that phloem sieve tube elements by osmosis, and it starts to increase in pressure at the top. Therefore, there is a hydrostatic pressure gradient, with the bottom having a lower pressure and the above having a higher pressure. What happens is the sucrose upwards moves down that hydrostatic pressure gradient from a higher pressure to a lower pressure. Therefore, the answer in this case is going to be B. Which are present in the walls of veins? Endothelium. This is correct. Endothelium is present in the walls of all blood vessels, including capillaries, arteries, and veins. Smooth muscle. Smooth muscle is also present. The role of smooth muscle is they contract to alter the volume of blood or the pressure in the veins. Elastic tissue. As we all know, elastic tissue stretches and recoils in order to control the blood pressure in the veins, and both arteries and veins have elastic tissue. Collagen fibers. All blood vessels do have collagen fibers for support. Therefore, the correct answer in this case is going to be D. True is correct for the location of a type of molecule or cell. Type of molecule: antibodies, blood, lymph, and tissue fluid. Here it's assuming that is not present in the blood. However, this is incorrect because in order for it to go into the lymph and tissue fluid, it has to be present in the blood in the first place. B. Plasma proteins are present in blood. This is correct. Lymph, tissue fluid. We cannot assume that plasma proteins are totally not present in lymph and tissue fluid. The reason for this is only the small plasma proteins are able to penetrate through the capillary, endothelium lining and go into the tissue fluid, then the lymph. So here we cannot assume that they are totally not present, so this is incorrect. C. Lymphocytes. Lymphocytes are present in the blood, this is correct, and present at tissue at the same time, so this is also correct. And the answer is going to be, what effect does decreasing carbon dioxide concentration have on hemoglobin? So first we must know what happens when increasing the carbon dioxide concentration, not decreasing. When carbon dioxide concentration increases, more carbon dioxide combines with water to form carbonic acid by the action of the enzyme carbonic anhydrase. Then this carbonic acid dissociates into hydrogen ions and bicarbonate ions. What this hydrogen ion does is that it binds to hemoglobin, forming hemoglobinic acid. And hemoglobinic acid has a lower affinity for oxygen, therefore oxygen is easily released. And hardly absorbed. Let's see the suggestions. A. It is less efficient at taking up oxygen and less efficient at releasing oxygen. Here we're looking at decreasing factors. This is incorrect. It's actually more efficient at taking up oxygen because it has a higher affinity. And then, less efficient at releasing oxygen. This is correct because there is less concentration of hydrogen ions, making it less efficient at releasing oxygen. Therefore, the answer is going to be C. 31. Why does the red blood cell count increase in humans as they start living at high altitude? A. To allow more carbon dioxide to be excreted by the lungs, preventing a fall in the blood pH. First of all, high or low altitude has nothing to do with the carbon dioxide concentration. It is not altered whether the person is at high or low altitude. B. To allow more oxygen to be taken up by the hemoglobin as the partial pressure of oxygen is lower. This is correct because the number of red blood cell increases to compensate for the lower partial pressure of the oxygen, to transport the exact same amount of oxygen as if it was in normal altitude, not high altitude. Therefore, B is the correct answer. Just a side note, the syllabus starting from 2022 onwards have actually deleted the altitude part, so it's no longer in the syllabus. 32. The diagram shows a photomicrograph of part of a bronchus. Which label identifies smooth muscle? First of all, let's identify these structures. Here, it's the pseudostratified columnar epithelial cells, and this is the epithelium found in the bronchus. So this is not a smooth muscle. C. Under that epithelial layer, you're going to find the smooth muscle looking parallel to each other, like that. So C is correct. A is actually the cartilage. And bronchus have blocks of cartilage, irregular blocks of cartilage. For trachea has C-shaped cartilage. So the correct answer is going to be C. Which statements about all bronchioles are correct? 1. They have epithelium. Bronchioles do have epithelium up until alveolus also does have epithelium. 2. They have goblet cells. This is incorrect. Only trachea and bronchus have goblet cells. They have muscle tissue. This is correct. The reason for this is to be able to control the volume of air in the lungs. So the answer is going to be B. 4. Which row shows the effects of emphysema? Emphysema is the decrease of surface area of the lungs due to the bursting of alveoli. So let's see the suggestions we have here. Here in alveoli, it never shrinks, it actually bursts. due to a decrease in elastin. Elastin is the elastic layer allowing alveoli to actually stretch without bursting. So if there is less concentration of elastin, alveoli will burst. Then number of capillaries. Due to the bursting alveoli, number of capillaries actually decrease. Therefore, the answer is going to be A. Is correct. Disease transmission, infectious or non-infectious. Let's start with HIV/AIDS. Here it's assuming that the transmission is genetic. This is incorrect. It's transmitted by a virus, human immunodeficiency virus. B. Lung cancer. It is not a pathogen. C. Sickle cell anemia. It is genetic. D. Tuberculosis. Here it's assuming that tuberculosis is non-infectious. Actually, this is incorrect. Tuberculosis is infectious and it is transmitted by airborne droplets. Therefore, this is incorrect. The only correct answer is going to be the diagrams show the results of incubating 2 species of bacteria, P and R, on separate petri dishes, each containing nutrient agar. 3 filter paper discs have been added to each petri dish. Which statement is correct? Antibiotics X and Y are resistant to bacteria B. There's no such thing as antibiotic is resistant to bacteria. It is the bacteria that's resistant to the antibiotic. Therefore, the statement is incorrect. Bacteria P are resistant to both antibiotics. This statement is actually incorrect. If bacteria P were resistant to both antibiotics, then they would actually grow around the antibiotics, just like bacteria R and X. But here, they've left a distance. This distance actually shows that the bacteria is not resistant to the antibiotic. Otherwise, it would normally grow around it. Therefore, B is incorrect. C. Bacteria R have a mutation that makes them resistant to antibiotic. Why? As we said, if the bacteria was actually resistant, it would grow around the antibiotic, not leave a distance. So this is also incorrect. D. N is a control disk showing bacteria R grow when bacteria Y is not present. This is actually correct. As we can see, it's present in both and it is a control disk. So D is going to be correct. 37. Statements are about infectious diseases. Which row matches the correct statement numbers with each disease? Let's start with A. Cholera. Disease may be spread between humans by contact. This is incorrect because cholera is transmitted by fecal, oral route, not by direct human contact. Therefore, 1 is going to be incorrect. Then 2 and 3, disease-causing organisms has many antigenic variations. This might be correct and might not. We're not certain of this information. 3, spread of disease may be influenced by climate. This is actually incorrect. The only disease that is influenced by climate is malaria because it has an insect vector which is female anopheles. Therefore, 3 is also incorrect. 4. A vaccination program is available. Yes, this is correct. There is a vaccine for cholera. Let's move on to measles. 1 and 2. Disease may be spread between humans by contact. This is correct. Because it's a virus, it spreads by airborne droplets. So 1 is actually correct. Let's see 2. Disease-causing organisms has many antigenic variation. This for sure is incorrect. The reason for this is that the reason why measles was eliminated in the first place was that it did not mutate often. And the virus stayed stable for a long period of time, so it did not mutate. This is what helped with the vaccination programs. So 2 is certainly incorrect. 4. A vaccination program is available. As we just mentioned, this is correct. 3. Spread of disease may be influenced by climate. This is also incorrect. Malaria. 1 and 3. As we said that malaria has a vector which is female Anophele mosquitoes, so it's not spread by direct human contact. so 1 is incorrect. For 2, disease-causing organism has many antigenic variation. Yes, because the pathogen is a eukaryote, it's Plasmodium and it's a eukaryote, it has many antigenic variations. So 2 is correct. 3, spread of disease may be influenced by climate. As you just said, that female anopheles prefer humid climates and climate which has puddles of water. Therefore, 3 is also going to be definitely correct. Let's see 4, a vaccination program is available. This is uncertain information because it is available but not necessarily successful. So the only correct answer in this case is going to be D. Muscle cells have cell surface receptors for neurotransmitter ACH. These receptors allow the muscle to respond to a nerve impulse. In the condition myasthenia gravis, the helper T cells stimulate a clone of B cells to become plasma cells and secrete antibodies that bind to and block these ACH receptors. The muscles cannot be stimulated and begin to break down. A short-term treatment for myasthenia gravis is to inject monoclonal antibodies into the blood. What is the target of these monoclonal antibodies? So, in basic words, there are antibodies produced against self-antigens directing the neuromuscular junction. So, in order to stop these self-antibodies binding to the neuromuscular junction, we have to actually stop them. How? By using monoclonal antibodies. So let's see the suggestion that we have here. Antibodies that bind to ACH receptors. This is totally correct because here antibodies are produced against self-antigens and we are trying to stop these faulty antibodies binding to the neuromuscular junction. So the only way is to produce a monoclonal antibody that's going to bind with that self-antibody to stop it binding to the neuromuscular junction receptor. So A is going to be the correct answer. 39. The diagram shows the simplified structure of an antibody. Which statement is correct? U and X show the antibody to bind to two different antigens. To start with, X and U are constant regions. And both of them has the exact same shape. Therefore, they cannot bind to two different antigens. They actually bind to the same type of antigen. Therefore, this is incorrect. B. V allows the antibody to fit around the antigen. V is the hinge region. And the hinge region gives flexibility for the antibody to allow it to fit around the antigen. So this is correct. C. W can bind to one specific antigen. First of all, W is the constant region and it does not bind to an antigen. It binds to a phagocyte or a receptor on a phagocyte or a macrophage, for example. So this is incorrect. D. X can bind with a specific phagocyte receptor. As we mentioned, it's W which binds to macrophages or phagocytes. It's not X. X is a constant region and it binds to antigens. Therefore, this is incorrect and the correct answer is going to be B. Question number 40. Why have vaccination programs eradicated smallpox but not tuberculosis? A. Vaccination can provide immunity only for smallpox but not tuberculosis. This is incorrect. Vaccination can actually provide immunity for both if done correctly. B. Only tuberculosis is associated with poor living conditions. This is incorrect. C. The smallpox virus showed little variation of antigens. This is correct, and this is the reason why smallpox was almost eradicated in the first place. If the antigen remains stable of a high period of time, this means that we don't need booster vaccines. Therefore, the vaccination program is more likely to be successful if no boosters are required. Therefore, the answer is going to be C. Thank you very much for watching this video. If you like this video, please share and subscribe.